Thank you. I've never been called a geneticist before, but I'm a chemist, so I apologize for bringing chemistry to this. So I'm going to talk to you about a platform that we've been developing with Joseph and the Seaver Center on trying to design small molecules that can target the wild-type allele of genes to boost their expression in autism diseases that are caused by haploinsufficiency. So I'm going to go through sort of how we view RNA-targeted small molecules and their potential. And so I have the reason that I picked this picture to start is I think we're now, this is tunnel view for Yosemite. So I torture my children every year, and it's coming up, to go to national parks. And I randomly took this picture. And so I think the sun now is rising on especially autism spectrum disorders to be able to understand targets and be able to hopefully develop precision medicines. And I want to start by thanking Joseph, Anna, Nan, who've been working in collaboration with our group, including Jessica, Aya, Liz, Nick, and Kisu. And I think it's really important to recognize the people that did the work here. You know, science is an international thing that we all have to work on. We have to bring diverse perspectives into that to do the best science we can. As I mentioned earlier, I'm a chemist sitting here in a genetics conference, and so that's just at a big level, but I think at a smaller, more local level, we have people like Kisu who come from South Korea to come to the United States to do science. And that's a special thing that we need to foster and recognize, as Joseph had said in the beginning of this. And then I am putting this slide at the beginning of all of my talks, and I want to push all of you to preach the Bible of science to your local communities. I'm one of 7 kids. I'm the only scientist of the 7 kids. And that means that sometimes at Thanksgiving, the conversations can get very interesting. Now, I didn't go to Thanksgiving this year. You can probably wonder why. Might not for the next few years. But nonetheless, I want to present on the slide that I had up there is that one thing that we can't doubt, regardless of whether we voted for one or we have a bias for one political party or another, is you cannot doubt that science has made our world a better place. And we have to communicate that to people. And we've made the internet, which for good and bad allows us to communicate things very quickly. We know genes that cause autism spectrum disorder and can make precision medicines. And so, and that's the backbone of all of that has been the US taxpayer. So we really need to be grateful for them for that, and not the moment we make science political, we lose. We cannot do it. So that's very critical that we all figure out how to communicate that to everybody. All right, so I am really an RNA physicist, and so I like to understand the way that RNA folds into shapes and controls biology. And so here's a very quick overview of, DNA goes to RNA, goes to protein. And we all consider knowing the genetic basis for diseases as making both coding and non-coding RNAs that can provide targetable structures to develop precision medicines. One of the biggest things that we found with the human genome being sequenced is that only about 1 to 2% of our genome gets made into protein, yet most of it gets made into RNA. And that's going to play a pretty important role in what I'm going to discuss in this talk. And as Joseph had said early on, is that when one considers addressable targets in the human genome, it's rarely thought of, at least in small molecule development, that RNA can be an addressable target. You guys can hear me in the back? It's a terrible question to ask. If I get no response, I know you can't hear me. And one of the molecules that I think has pushed forward the hypothesis that RNA can be drugged with small molecules is this compound called risdiplam, which is the third, but I think the most powerful medicine for treating patients that have spinal muscular atrophy. And how that molecule works, it's going to come into the end of our talk, is that it binds to and stabilizes RNA-protein interactions to affect inclusion of to affect inclusion of an exon to convert SMN2 protein into a longer SMN2 mRNA into a version that can encode a longer lasting protein to substitute for the loss of survival motor neuron 1 protein in children that have spinal muscular atrophy. Let's see. And so really the way that RNA plays a role in diseases is it plays both coding and non-coding functions, and we should consider each of these as being addressable targets. So really what we're trying to do here is develop an understanding for what are the regulatory domains in RNA that one could target with an orally bioavailable small molecule to, in the case for autism spectrum disorders, increase the expression of the wild type allele to substitute for the loss that happens due to haploinsufficiency. And so that's what I'm going to go through here, and we've built a platform to be able to do this broadly to study RNA targets, but later on I'm going to talk about some lead chemical matter and compounds that are associated with these targets as we push forward. And I'm going to talk about 4 mutated genes that we've targeted, some that work to boost the wild type expression, a wide variety of cancer, to also, as Joseph had mentioned in his teaser slide, patient-derived cells. And then, but our overall goal is to develop oral compounds that can cross the blood-brain barrier to be able to do this across a wide variety of diseases. Now, here's a flavor very quickly for how small molecules that target RNA can work through the lab. We can decrease protein production, so we've been able to design and deliver molecules that can target basically ALS causing genes if they're repeat expansions, and C9orf72 to decrease protein produced. This is an RNA structure that regulates translation of alpha-synuclein protein, and so this RNA structure that's the type letters here, actually if you target that with a small molecule, you can decrease the production of alpha-synuclein. You can also target RNAs to affect gene expression or to affect the identity of the protein that emerges from a messenger RNA. This is an RNA structure that's in the microtubule-associated protein tau, and if that structure is mutated, you get too much of 4R tau. Now, the reason that I put this in here is that we have this paper on bioarchives where we can take these molecules, these lead tool compounds that we have, and we can convert them using medicinal chemistry into orally blood-brain penetrant molecules. And that's demonstrated with this tau molecule that lowers the toxic form of tau. And we think this platform, once we identify the sites that compounds bind on RNA targets, is a general one where we can start to build up potency in compounds to get them to be, again, oral blood-brain barrier penetrant to affect disease-associated pathways. Now, as I mentioned earlier, we're in an ASD symposium here, and many of these diseases are caused by haploinsufficiency. And so what we really want to do is not change the protein that emerges from an mRNA or diminish it, is you want to actually increase mRNA protein production from a given messenger RNA. And we've done this a lot by targeting microRNAs. But microRNAs, which earned, I hate the term won a Nobel Prize, earned a Nobel Prize this year in medicine, are these small RNAs that are made from precursors which bind to 3' untranslated regions of messenger RNAs, and they turn down the amount of protein that's made. And so we've been able to develop a wide variety of small molecules that can inhibit the production of the translational inhibitor to increase protein expression. But one of the issues that we have in microRNA targeting is their recognition element in messenger RNAs is only a small number of nucleotides. And so what they do, and I do think this strategy could work for ASD, is that it increases the expression of pathways of certain genes. And oftentimes that's regulated in cancer, but that's a mechanism that we could work on also in these diseases. But what we've been focusing on is the 3' untranslated region of RNAs in these, in the wild type allele of ASD-associated genes. And it turns out the 3' untranslated regions of messenger RNAs and the 5' untranslated regions of messenger RNAs tend to be very well structured, and those structures within those targets can endow these transcripts with the ability to bind small molecules. I'm going to focus on the 3' untranslated region of these diseases because there's a lot of regulation that occurs in terms of increasing translation at the 3' end, which can include stripping off or augmenting the binding of RNA binding proteins to the 3' end. It's where poly-A adenylation or poly-A tails can be increased on these targets. And so if we can modulate the 3' end of messenger RNAs with small molecules, then we can increase translation, we think, in a pretty robust way. So just very quickly, 2 ways in which the compounds can work is that they can confer stability on a messenger RNA by stabilizing the binding of proteins to those mRNAs, which could increase translation, or these molecules could basically inhibit the binding of RNA binding proteins where the RNA binding protein could diminish the poly-A tail length of a messenger RNA. Okay, so here's the platform that we've been developing with the Seaver Center, Joseph and Anna and Nan, where what we've been doing is taking all of the genes that are haploinsufficient in autism spectrum disorder, and we've been looking at the structure of these 3' UTRs. And what we're doing is we're looking at all of, because in these diseases and in human populations, you get varying different RNA forms. So you get varying isoforms at the 3' UTR. And what we do is we analyze all of the isoforms that we see in a given patient population to try to understand what are the structures within these different forms of the mRNA. And we make a modified construct where we take the 3' UTR, that the structure that is harbored into it represents the most common form of RNA structure through all transcript variants. And what we do with that, can you go back for a second? I'm sorry. Yeah, and so what we do with that is we identify 3' untranslated region targets that are structured, conserved, and protein bound, and shared across transcript variants. And so we've looked at a wide variety of targets. Here are the 4 main targets that we looked at: ADNP, FOXP1, DDX3X, and SHANK3. And I'm going to talk to you quickly about some of the compounds that we've been able to identify that can increase in both model and, as Joseph gave you a teaser for, non-model systems, the expression of these proteins. This is early stage compound development, but one of the things that we want to do in this is we wanted to make sure that the early stage work that we did was done in a manner that's consistent, where at the end of the day we can deliver an oral blood-brain penetrant compound that we can assess across multiple different scaffolds to see which compounds are effective, safe, and that we can, again, progress into the clinic. And so what we did is, again, we took and fused to luciferase these 3' UTRs across these genes that express the most common RNA structure across variants. And what we did is we screened those reporters for binding to a library of small molecules that are focused for binding RNA. The reason that we looked at those compounds, and it's between 2,500 compounds, but we've, over the course of many years, been able to annotate a library of molecules that are enriched in RNA binders quite dramatically. Now you can ask, why did you want to do that? Well, our mode of action, given that what we just talked about is binding to RNA targets, and one of the issues, is, which if you go through the history of Risdiplam and Branaplam, a Novartis compound that ultimately didn't get FDA approved, is that when you typically take molecules that regulate RNA processing in phenotypic or binding screens, oftentimes the hit rate that you get with RNA is very low. And the challenge with that is it's very hard to be able to pick out compounds that you can then take and invest medicinal chemistry resources in to optimize them. Because if you only get one hit from a scaffold series or one hit from a compound class that's active, it's very hard to make decisions about which one of the many compounds you have to be able to push forward. Ideally, you'd like many compounds across a scaffold hit to be bioactive that you could then address and make decisions. So I'm going to tell you high level about some structures here within this, and then you can make some decisions about the utility. But we have a wide variety of informatics capabilities where we can not only identify ligands that have a high probability to bind RNA that can increase our hit rate, but we also have, based on these data, the ability to very quickly optimize these molecules, as has been shown with this tau compound that's on bioarchives. And so here's basically what we did with this screening approach. So there's some complications in this in the Venn diagram, but basically what we did is, again, we took these luciferase constructs where we have the most common RNA structure form in the 3' UTR, and we screened this RNA focus library across binding to all of these 5 reporters. So it's an ADNP, a DDX3X, Shank, and FOXP1 reporter. And then in the presence of luciferase. And basically what we did is we threw out compounds that overlapped with any of these targets. Now you can make a comment and say, well, Matt, it would be nice if you had one compound that could boost the expression of all of these. I'd say that's a pretty high bar, and I don't want to have that be a complexity as we're trying to lead optimize molecules. And so what we did is we identified molecules that specifically could boost in these model and also in less model systems specifically. And what we were able to find is that for each of these mRNAs, which is consistent with, we think, the molecules achieving selectivity, is that we have different compound scaffolds that emerged for each of these targets, which we've seen previously when we screened small molecules binding RNA. So I'm going to show you very rapid fire some of the structures and activities of these compounds as we go on, and then you get a feel for what we're doing with Joseph and the Seaver Center team. At the end of the day, we want to simplify this to a small molecule drug discovery platform. We want to use small molecules that are oral blood-brain penetrant to be able to intervene with these RNA targets to boost the expression of the wild-type allele. And one of the other advantages that we have here is that in contrast to, say, I would say a gene therapy approach where you could have massive increase in the expression of some of these targets, is that our expression levels is between 50 or 100%. So because we're interfering in the natural way in which these RNAs are processed to increase translation, it's unlikely that we get this massive boost in the expression that would have issues. And because it's oral, we should be able to deliver an increase in expression depending on where the molecule goes, which could be part of the medicinal chemistry pipeline. So here's just some of the scaffolds that we have. Here's the ADNP preliminary SAR. You can see these molecules have these types of structure-activity relationships that we get within them. We have a wide variety of molecules that we can do chemical synthesis to be able to get these compounds to be bioactive. And here's just some of the SAR that we have. And then those will boost in the model system. Here's the follow-on what Joseph had said, and what I'm going to show you is more sort of compounds, is here's the ADNP expression if we go into HEC293 T cells, which is our model before we go into more patient-derived cell line. You can see that these compounds can increase the expression of ADNP in these cells. So we can translate molecules that work from this luciferase reporter all the way to compounds that increase the expression in these wild-type cells and furthermore into patient-derived cell lines. The reason that we're showing some of these are toll compounds. You can see they're very, if you're a chemist, they're very low molecular weight compounds that have room for significant optimization, and we're able to get activity of these compounds with these starting tools in low micromolar concentrations. Here's just more data for FOXP1. We have, again, a different chemical scaffold where we can then do file mining to have other compounds that have activity. And again, we're just sort of showing that we have optionality within these targets. It's not one scaffold that's boosting all of them, which I think we would probably deprioritize, at least at this beginning stage, is that there's scaffold diversity within these compounds that are working across the targets. And these molecules have very low molecular weight, which suggests that they should be oral blood-brain barrier penetrant. So I'm just going to go quickly. Here's more SAR within these targets. And so at the end of the day, we have bioactivity in model systems against all 4 of these genes by using this platform. And what our next steps in doing this is we're doing in vitro PK, where we're studying like normal small molecule developmental pathways do. We're studying the stability of these compounds and their permeability. Based on the selection of the molecules that we did, we have the ability to make 100 of these compounds in a day, and that will allow us to very rapidly construct what is the best compound that's more potent. We have to get these compounds below sort of the 1 micromolar activity data. We need to drop the potency between 10 or maybe 100-fold within this, and the ability to make many compounds to do that is very important. And we also have a program here in both the model cellular systems that we have, but also the patient-derived cell systems to really understand mode of action for these compounds. One thing that we're doing in support of that mode of action work is we have a wide variety of, I'm almost done. I told you I was gonna give you 5 minutes back. So I'm just gonna pause and then take the 5 minutes. I went to Catholic school, so I don't follow instructions. So one of the things that we can do within these scaffolds is we have the ability to append molecules that can cross-link with both protein and RNA targets in cells. And here's just a prototype molecule. So if you can throw things in cells and cross-link them, you can pull them down and then run RNA-seq or proteomics to be able to identify what are the RNA binding proteins that sit on the 3' UTRs of these messenger RNAs. Do these molecules cross-link with the messenger RNAs, or do they do something else, which can allow us to then do modeling to really beef up the potency and the activity of these compounds. And then one of the other things that we've done, as Joseph had mentioned earlier, is we're testing them in patient-derived cells, including neurons, and that allows us to run RNA-seq and proteomic profiles for each of these compounds as we go through a developmental pipeline. One of the reasons that we used phenotypic screening up front to be able to look at basically 3' UTRs is it's very similar to the development pathway that Risdiplam had. And we have the ability, after we do phenotypic screening, to do cross-linking to try to identify or at least deconvolute the RNA targets that these molecules have. And as I mentioned earlier, this is a platform. I have to tell you, I was pretty surprised that we were able to get scaffold diversity within these compounds that can increase protein expression. And so we've identified with Joseph and Anna about 130 priority mRNA targets where we think that there's an RNA structure in the 3' UTR, and we've identified a sequence that houses most of the transcript variants and the functionality. And so we have the ability to do broader screening here. And so the question that we're going to have going forward as we expand this platform, I think it's very powerful to do screening across many targets because it tells you what are frequent hitters and the way that meaning compounds that are active against multiple pathways, and we don't want those. So this is going to allow us, as we expand the platform and the chemistry that's developed in this, to try to identify, compounds that we have the best chance of being able to develop. Because at the end of the day, molecules need to be safe and effective in these patients. And I think this is a huge unmet medical need. That's one of the beautiful things I think of RNA, is that you can increase the expression of targets, not just by binding 3' UTRs, but also by binding these microRNAs to increase pathway expression. And so I want to end here by thanking you guys for your attention during this and thank the people that did it. I also thank the US taxpayer, right? So we don't do that enough. They're really partners, and I know many of you might not be from the United States, so I guess I should say global taxpayer. We need to thank them for enabling us to do the science. Not only that affects their families, which the Seaver Center knows all well, but actually families in their communities. If we didn't have the resources for that, we wouldn't be able to do what we do and really do the science that we all dream about. It's pretty amazing to be able to have an idea and then come into a lab and work with people and test that idea the next day. That sure beats working for a living. So thank you for your attention.